Today we have a presentation by Matthew Medeiros. Medeiros is a researcher with the University of Hawaii. His presentation is titled Environmental and Ecological Factors Influence the Gut Microbiome of Wild Picture-winged Drosophila across the Hawaiian Islands. His research is conducted through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. Matthew, you may start whenever you're ready. Mahalo.
All right, thank you, Tristan. And to those of you who are attending right now, and actually I'll thank anybody who may eventually see this on the YouTube channel as well. Okay, hopefully my presentation is visible now.
Looks good. Yep.
Okay, thank you, Tristan. Okay. So yeah, I oftentimes go by Vegas Matt because I am actually in Las Vegas, Nevada right now. And I go by Vegas Matt to differentiate myself from Oahu Matt, who is also named Matt Medeiros and whose office is right next to my lab. And we get confused all the time. I'd also like to thank Joanne Yu and Don Price, who are my co-primary investigators for this research. Joanne Yu is my advisor at the University of Hawaii Manoa, and Don Price at UNLV. And I'm also going to be talking about contributions from Sean Scoville from the University of Wisconsin-Madison, who has helped a lot with some of the analyses for this project. This is what it looks like right outside my window right now, of course. And this is what my evening last night looked like, but I got some sleep, and I'm here now to tell you about the Hawaiian picture-winged Drosophila. So this is a picture of Drosophila grimshawi taken by Carl Minyaka, who's done a lot of great work on the Hawaiian picture-winged Drosophila. The Hawaiian picture-winged Drosophila are these incredible little flies. You can see that they have these amazing wing patterns. This particular species has a lot of pigmentation on its wings. Some of them have more or less. And this fly is actually rather large for a Drosophila. Some of you who have heard of Drosophila melanogaster, let's say that's a really common lab organism. Drosophila melanogaster is only about 1/10 the size of this fly right here. So these Hawaiian Drosophila are really quite impressive, not only in terms of their coloration, but also their size as far as compared to the rest of the Drosophila in the world. These are some other examples of some types of wing pattern that are in some of those Hawaiian Drosophila. So there was some long time ago, a continental species of Drosophila that somehow got to Hawaii, maybe blown over in a storm or on a mat of vegetation that sometimes gets swept out to sea during, say, tsunamis or earthquakes. And oftentimes larvae of insects will then be able to get to new places this way. And those larvae can grow up and they can become separated from their founding population. And they can then spread around to other Hawaiian islands in this case. And then they can diversify, like we see in this illustration here from Edwards and others back in 2007, showing just some of the diversity of wing patterns in these flies. How many Hawaiian Drosophila are there? It turns out that there are hundreds of species of Hawaiian Drosophila. These are just some of the species. They're obviously way too small to read in this recent phylogeny. But this just does give you an idea of how many species have resulted from that apparently one colonization event to Hawaii, something on the order of 20+ million years ago. So going back a slide, some of the Hawaiian Drosophila are not what we call picture-winged Drosophila, but many of them are. And so the ones that I'll be focusing on in this talk for the most part are these picture wings, which just make up some small part of that overall family tree of Hawaiian Drosophila. And these are probably the best studied Hawaiian Drosophila. They may even be the best studied Hawaiian insects. They're really interesting for a number of reasons, not only their large size and their amazing wing patterns, but they also in many cases are specific to one type of host plant. And so in this phylogeny, Minyaka and Price 2015, you can see the different lineages of these species, and we have the various Drosophila species up here, picture-winged Drosophila species. Various species of those have eaten a number of different, evolved to eat a number of different types of host plants. So they're really quite amazing because in many insect groups, we don't see nearly this much diversification of what they eat. It turns out it's pretty tough for an insect to start eating something new. It doesn't oftentimes happen that often. But in the case of the picture-winged Drosophila, it has happened a lot. So all of the facts that I've just told you about picture-winged Drosophila, they're a crazy diversification. People have tried to explain this for a really long time in Hawaiian biogeography. For well over 100 years now, people have been exploring these flies and seeing what we can learn about biology in general. And some of the main hypotheses for how this lineage got so diverse, why there are so many species, first of all, allopatry, or geographical separation. That's when we have lineages where certain members of that lineage move to a new place. Now they're separated. Evolution will presumably work on them differently, whether it's natural selection or genetic drift to cause differences. Sexual selection may also be important in these flies. They have oftentimes really elaborate mating rituals, especially the males. And so it could be that those behaviors then either attract or repel other females in certain cases, which could then cause lineages to split as well. And then thinking back to this slide right here, we've had all these host plant changes. And if flies are on different host plants, then perhaps that's one additional reason that these things have diversified so much, because if they're found on different plants, then presumably they're not coming into contact with each other as much as if they were on the same group of plants. That said, host plants, oftentimes we're talking about the larvae, and we all know that baby flies, the larvae of flies, we call those maggots, not necessarily the cutest little creatures for many of us, but nonetheless, those immatures, those larvae, the maggots, eating different host plants may help to prevent lineages from mixing, and in fact may increase the rate of diversification in these flies. But a fly is not actually just a fly. A fly is the fly, plus it's all of the other organisms that live in and on the fly, such as bacteria and fungi. And those are going to be one of the big focuses of my talk today, is how do these other creatures that live on the fly affect potentially its diversification. It's not just flies that are holobionts. Let's think about ourselves for a moment. You may think of yourself as yourself, and you are probably yourself, but you are also an assemblage of many other organisms as well. We have, in many cases, heard about those little mites that live on our face, for example, and crawl around at night when we're sleeping. Our skin, of course, and our guts are filled with bacteria. We have fungi on various parts of our body and in various parts of our bodies as well. So this assemblage, you can think of yourself as an assemblage of organisms, your own organism plus all these other things that live in you and on you. And you can think of yourself as there then being a host, and the host plus those organisms being what we call a holobiont. And this way of thinking about organisms as holobionts, or as actually assemblages of organisms, has been really helpful in recent years when trying to learn about a whole slew of characteristics of organisms. For example, humans, we actually survive much better if we have certain types of bacteria in our gut that help us digest food or to make neurotransmitters. They can make our skin in much better condition, or conversely, much worse condition. When we take things like antibiotics, it obviously has some really important pros, but it also has some cons as well. Oftentimes messes up our digestion, for example. So there's been a huge interest in recent years as we've developed techniques to study bacteria and fungi in us and on us to try and figure out how these things are really affecting us, how the whole holobiont works as one major organism. And I'll also say that it's pretty interesting to note that in us and on us, as far as humans go, we certainly have way more bacteria and fungal organisms in us and on us than our own cells, than our own organisms. So that's a pretty interesting thing to think about. So now that we have the holobiont concept in our heads, there are some additional ways that picture-winged Drosophila might be diversifying in the Hawaiian Islands. Perhaps they are landing with new or obtaining new microbes in their gut to help them detoxify plants that they're eating. And if they are eating different plants and getting different gut microbes that are on those plants that help digest those plants, then perhaps those gut microbes are leading to changes in what we call cuticular hydrocarbons, or CHCs. Cuticular hydrocarbons are the waxes that are on the surface of essentially all adult insects. And those cuticular hydrocarbons include things like sex pheromones, which allow flies to find each other, to find mates of the same species. So to go back a little bit, we have these traditional hypotheses for diversification of the picture wings. But now we have this new set of hypotheses for the picture wing Drosophila that all stem from the fact that we know that they have these bacteria and fungi in them, and that those might be leading to changes in the flies. So before I get into some of the field work that I've done here, a little bit of background is that it turns out that altering the gut microbes in these flies does actually change them in some ways that may be important for diversification. So this is from a paper with Joanne Yu, Don Price, Loris, and Aziel Macias, and myself. We all worked together on this, a paper that's currently being reviewed. And in this paper, we looked at how gut microbes affect flies in the lab. And what we did is we actually knocked out either bacteria or fungi or both from the guts of the flies in our lab. And it was the same species that I was showing up here, Drosophila grimshawi. And it turns out that, for example, if you knock out the gut fungi in female flies, they actually have very few matings. So if we look at the bars over here on the right, for example, we can see that most females that have no gut fungi had no copulations at all. And then other combinations, like when females did have gut fungi, there was not a problem in terms of number of copulations. Those flies copulated about as much as control flies. So it's certainly possible that changing the microbes affects the will to reproduce for some of these flies. That may be because it turns out, and I don't have a picture of this on this slide, but females that don't have enough gut fungi actually don't make enough eggs. So it seems like the females know that they're not making eggs because they don't have the proper microbes in their gut, and therefore they're unwilling to mate. If we look over at the left, this is that term again, CHC, or cuticular hydrocarbon. And these cuticular hydrocarbons actually change in quantity when we knock out, again, the gut fungi. So this is flies without gut fungi over here. AF stands for antifungal. And we can see that males, for example, have a statistically significant increase in pheromones or cuticular hydrocarbons when they've had their gut fungi knocked out. So this is kind of like a little background proof of concept that change in the gut microbes of flies really does change their behaviors. So then what we wanted to do was we wanted to ask the question, do fly gut microbes actually differ in nature between different fly species and in different locations? We know that changing the microbes in the lab can cause differences in reproduction, but do we see differences in nature? If so, then perhaps this would be one hypothesis for how the picture wing Drosophila have become so hyper diverse. So our idea here was to go out into various forests in Hawaii and collect picture wing Drosophila. And here you can see my wife and very able field assistant, Ann Nguyen. She is a pediatrician by training, but she really enjoys catching picture wing Drosophila whenever possible. And here we are actually at Hawaii Volcanoes National Park on the Big Island. And Anne is looking for flies here, and it's a little maybe hard to see, but in this picture there are some sponges. You can see kind of the back of a green sponge hanging from a tree. The way we catch the flies is we put rotten mushroom fermented water onto the sponges using a spray bottle, as well as some fermenting banana with some yeast in it. We put that onto the sponges as well, and the flies become obsessed with it. They love the smell. They will land on the sponges, and then we can just pop them into a little vial and take them back to the lab for further exploration. What did we do with them back into the lab? Well, at this point, sadly, the fly dies. We kill it. We surface sterilize it with ethanol and bleach. And then we put the entire fly, after grinding it up, into a DNA sequencer. And we will not only get DNA sequences of the fly back, and this picture here on the top right is just kind of what I see on my end, the different DNA sequences, but we also get DNA sequences for bacteria and for fungi. we sequence a type of a gene for that's called 16S ribosomal RNA gene. That's a really good way of finding out what type of bacteria you have in your sample. And then something called an internal transcribed spacer, or ITS. This allows us to identify what sort of fungi are in the fly gut. So this is kind of the entire methodological pipeline from collecting flies out into the wild to then seeing what their bacterial and fungal sequences are later on. Now, where did we collect flies? We collected flies from 6 different islands, including some that are not oftentimes collected on for this sort of work, which I'm pretty excited about. We got flies from Lanai and Molokai, as well as Kauai, Oahu, Maui, and the Big Island. And the stars represent some of the main places that we looked for flies and caught flies. In the lower left here is a picture of Joanne Yu and myself on a very rainy day in the Alakai Swamp at Koke'e State Park, or near Koke'e State Park, near the summit of Kauai. And then the other picture here is me with Don Price, and there we are also we are in Koke'e State Park on Kauai, but on a much more pleasant day. So we did a bunch of field work. We got as many different species as possible. And some of our field work was at the Laupahoehoe section of the HETF. So we're very appreciative of the HETF to allow us to be there and sample what flies were there. Here's one more picture of Don, Joanne, and myself. This is Hawaii Volcanoes National Park on the Big Island. And since many of us enjoy pictures, here's one more. This is my trusty brother, Dr. David Medeiros, who is a linguistics professor at California State University, Northridge in Los Angeles. And I dragged him out of his warm bed for about a week and made him sleep in a rat and mold infested lean-to near the summit of Molokai. But he was a very good sport, as you can probably tell from this picture. What did we end up with after all of these field surveys? We ended up with 30 different species of picture-wing Drosophila from around the Hawaiian Islands, as well as some other native and non-native Drosophila that are not in that picture-wing group. And we ended up with thousands of different kinds of microbes, so we could all analyze these data together. Now that I'm talking about data analysis, I will introduce you to Sean Scoville from the University of Wisconsin-Madison. Sean and I went to grad school together at UC Berkeley. He's a very good friend of mine, and we also collaborate together, usually studying these crazy organisms up here on the top right. This is a gorilla-bladed, or an ice crawler, a very seldom encountered type of insect. They only live on ice fields, and they're nocturnal, so they're pretty hard to get to. And down here are some pictures of Sean and myself in Yosemite National Park in the backcountry, waiting around for it to get dark so that we could get some gorilla-bladeds from the snow. It's pretty fun field work, pretty difficult, but pretty awesome. So Sean is really good at some of the analyses that I'm about to show you, much better than I am. So I do want to give him credit. He'll be a co-author on the paper that we're working on right now. To give you an idea of how the analyses work when we're looking at a holobiont, when we're looking at an organism plus its gut microbes, this is essentially the sorts of things, the sorts of like data that we're looking at. The top left is a file we get from the sequencer, from the DNA sequencing apparatus. We get these files. So the one on the top left here, this is a fly right here. This code is a fly. And then it's indicating to us that there were 372 of bacterial species 1, 531 of bacterial species 2, 10 of bacterial species number 3, and so on and so forth. We end up with thousands usually of bacterial species for any given fly. And this is just how many were in that particular fly's gut. This second column here is another fly. Okay, then I have this second spreadsheet right here. This fly right here, which is, I guess it's not this one, doesn't matter, but they all do match up somewhere in the spreadsheet. So like this fly right here, it was found on Kauai at Koke'e State Park. It was Drosophila velocipedis, one of the picture wings. So now the columns in this spreadsheet match up with the rows in this spreadsheet. We get a third spreadsheet from the sequencer that has the identity of that first bacterial species. So bacterial species 1 was in this genus, bacterial species 2 is in this genus, and so on and so forth. So we take those 3 spreadsheets and we use software, a computer language called R, and some of the different software analysis packages that are available to download from R. We basically overlay those three spreadsheets over each other, and then we're able to do a number of analyses using R. For example, here I have an analysis that's showing the number of different bacterial species for certain, maybe fungal species there for flies from different subclades of the overall Hawaiian Drosophila. So now I'm going to go through some of the outputs of some of these analyses. First of all, we asked the question, was there phylosymbiosis in these flies? So let's just look at the left side of the slide right now. Phylosymbiosis is the hypothesis that a holobiont, that their DNA, like in this case, the fly DNA, is essentially going to match the type of bacteria or fungi that are in their gut, and that they're going to evolve in parallel. So we could think about like species of fly, like 1, 2, 3, 4 species of fly. If species A has a really similar bacterial community to a certain bacterial community, and then its closest relative has another really closely related bacterial community, and then the next closest relative of fly has the next closest related bacterial community, and so on down the line. That's called phylosymbiosis. And that would suggest, if we see a pattern of phylosymbiosis, that the main reason that we have certain bacteria in our gut is because we evolved to have those bacteria in our gut, that those bacteria sort of change as we change. So we did an analysis of phylosymbiosis over here for the left side, and we actually did not see a strong pattern of phylosymbiosis. It turns out here there are fly species on the bottom and bacterial groups on the left, and we're just seeing like dark and light colors scattered throughout this this diagram. If we saw like bands of color that were similar to each other, then that would indicate that bacterial communities between fly species were similar, but we don't see any pattern like that. So it does not seem like flies have their gut bacteria or their gut fungi because of how they evolved. It seems like there's some other reason why they have the particular microbial communities in their guts. Looking at this figure on the right, the one with some fly species either in green or in black, we did a test to see whether these flies have a microbe, a bacterium called Wolbachia in them. And Wolbachia is a microbe that causes mating incompatibility between a lot of different insect species. And if we saw a lot of Wolbachia in closely related fly species, well, if we saw like every other fly species, one with Wolbachia, one without, one with, one without, then that would indicate that Wolbachia infection might be causing closely related flies to diversify. But instead, we tend to see Wolbachia clumped together in closely related species throughout this tree. So we don't think that Wolbachia is a main reason for the diversification of the Hawaiian picture wings. So what does affect what sorts of gut microbes are in these flies? We looked at a lot of different environmental conditions around the Hawaiian Islands, like what elevation we caught a fly at, or how much evapotranspiration there was. That measures like rate of water loss from plants as well as evaporation from the soil. And we also looked at how much it rains in various locations throughout the Hawaiian Islands. And it turns out that these actually do have a big factor or play a big role in terms of what gut microbes we expect in a fly. So the plots right here are for elevation. And this isn't telling us exactly what's in the flies at different elevations, but if you look down here where it says significance 0.001, that's a really small number. And that's basically telling us that statistically, we are seeing certain groups of bacteria and fungi in flies from certain elevations that differ from flies at other elevations. So we did get these really statistically significant values for elevation, transpiration, evapotranspiration, and rainfall. So it seems like, as I already mentioned, who a fly is closely related to doesn't really seem to affect their gut microbes much. What seems to affect their gut microbes is the habitat that they're in. So we think that the gut microbes are getting picked up by the flies from the plants that they're on. But this is also kind of interesting because when we sequenced, in this case, fungi from plants that these flies feed on a lot, such as Claremontia, which we can see over here on the left, and we looked at what orders of fungi are on those plants, versus the orders of fungi that are in our fly guts, especially the flies that are known to eat Claremontia. It turns out that there were only a few orders that were in common between those different things, the plants and the flies. They have very few fungi in common, which is pretty interesting because the flies must be getting their fungi from the plants that they're eating. But it seems like the flies are picking up only specific microbes and then like harboring those specific microbes in their guts. Maybe those particular fungi help the flies digest plant material, just like we use a lot of the microbes in our guts to digest our food. And it could be that these particular types of fungi also help the flies in terms of reproduction. Maybe it helps them make their pheromones. Maybe it helps them develop eggs, something like that. We're not sure yet what's important about these particular orders. But what is interesting, just to reiterate, is that the flies don't have very large abundances of many of the different fungi that are found on the plants. And we're also seeing the very same pattern in bacteria, that fly gut bacteria also really differ from what's on the plants that they're hanging out on. As many of you are probably aware, there are also a lot of non-native species in Hawaii, plants, animals, probably microbes as well. We don't know much about that. But we did collect some non-native Hawaiian Drosophila. And so we looked at the gut microbial communities for native versus non-native flies. And we actually saw that they were different. So this is pretty interesting. It turns out that native flies and non-native flies have significantly different abundances of a number of different bacteria and fungi, even though they're living in the same place. It could be because they're eating different foods, but it could be because their guts have evolved to have basically preferential treatment for some microbial species. We investigated this a little bit further to see how microbes change when a particular species of fly moves to Hawaii. This is a really prevalent invasive species in Hawaii, Drosophila suzukii over here on the right, or the two-spotted Drosophila. And it turns out that when Drosophila suzukii moves from other populations to Hawaii, it also has huge changes in its gut microbes. So these are now looking at bacteria instead of fungi. Over here on the left, these are some of the most common bacterial groups, genera in this case, from Drosophila suzukii in their native range of China. Over here are the most common ones in Drosophila suzukii collected in Hawaii. And then in the middle are bacterial genera that are in common to both. So kind of like we saw with native Drosophila harboring certain fungi from plants, turns out that the Drosophila suzukii really have only a few species in common. So maybe these Pseudomonas and Sphingobacterium are really important in some way for the Drosophila suzukii, and then they're getting like really different stuff maybe from the plants that they're in. This may be a clue as to how species change a lot historically when they get to Hawaii in the first place. Going back to that concept that if you move to a new area, you get new gut microbes, and this could then really affect your organism, your holobiont in a certain way, maybe in terms of mating and compatibility. We're doing some other analyses right now, and when I say we, I mean Sean. Sean has done a great job looking at some of the other aspects of how gut microbes might affect organisms. So this is just I'm not going to go into detail here, mostly because this is all on Sean, and we'll talk about this in the paper that we're working on. But Sean was breaking down host plant generalists versus host plant specialists, flies that just eat one plant when they're a baby versus flies that eat, that would be a specialist, or flies that eat many different plants when they're a baby, that would be a generalist. It turns out that they have gut microbes in many cases that do different functions. So this is talking about like different metabolic functions that flies might be doing in their bodies. This is just an enlargement of a part down here. So I'm excited to see what sorts of analyses Sean does with these data. All right, so I'm getting close here to wrapping things up. Changes in the gut microbiome of lab flies, as a reminder, do result in phenotypic changes. In this case, changes in cuticular hydrocarbons, changes in the amount of eggs produced, and changes in how many times flies copulate. And now we have seen that wild flies do indeed have different gut microbiomes in different locations between different species. So it may be the case that gut microbes maybe one cause of diversification, and maybe even an important cause of diversification in the Hawaiian picture-wing Drosophila. And this would really be a cool breakthrough, in my opinion, because there has been a lot of debate over the decades as people have been studying the Hawaiian picture-wing Drosophila, like what is leading to their diversification. And I certainly don't discount those other aspects, geographical separation, sexual selection, being really important in these flies, and host plant shifts. But I do think that these microbes are really important for their diversification as well. And I will just also mention that at the Laupahoehoe section of the Hawaiʻi Experimental Tropical Forest, we see at least 4 species of picture wings. Pretty commonly, we see Drosophila sprodi and Drosophila murphyi, and then less frequently, but still somewhat present some of the time, Drosophila sylvestris and Drosophila basicidi. These are actually all pictures by Carl Maniaka taken on various colors of supermarket sponges, the kind we use in our kitchens, and you can see this fermented banana on some of the sponges. With that, I would like to thank a whole slew of people who have helped me in the field, a bunch of different organizations that have helped me with permits and access to be able to collect these different flies, and then a number of different institutions for funding this research. And of course, I would like to thank Moki the Ragdoll legend, my ragdoll cat, who is slinking around my apartment even as we speak. And with that, I am happy to answer any questions folks may have.
Mahalo, Matt. That was fantastic. I don't see any questions in the chat, but I do have a couple questions for you myself.
Okay.
I'm curious, one thing, like how did you folks come up with the baiting with the banana and the mushrooms? Like how was that discovered as an effective lure?
Yeah, that's a great question. So what I don't know entirely how that, I did not come up with that, and nobody that I work with came up with that. That's been a method that's been used for a long time. It turns out, okay, so to back up a little bit, the first Western entomologists in Hawaii were some folks sent over by the British Museum of Natural History in the late 1800s. And what they did in order to collect these picture-wing Drosophila, they found that if they put kitchen scraps out in the forest, that the flies would actually come to those kitchen scraps. And it probably was an accidental discovery. They were camping a lot of the time in remote locations for weeks at a time. And so they probably saw these flies just coming to their food scraps. Later Hawaiian entomologists, like in the 1950s, when folks like Zimmerman and Sweezy, which are some big names in Hawaiian entomology, when they were doing their work, what they would actually do is they would take their kitchen garbage cans and they would lug them up into the forest, and they'd see that the flies loved the garbage. There's something about like fermenting vegetation that these flies are obsessed with. And so I guess from then people just were like, this is disgusting and kind of a pain, so we're gonna just use a subset of the garbage. I do know that Drosophila flies in general like the smell of rotting things, and so why it's the mushrooms and the bananas specifically that we use, I don't know. And it is possible that we're missing some species of flies that maybe don't like the smell of those things. But all I can say is that that's what people have been using for a couple decades now. And I will also tell you that the mushrooms in particular smell awful to humans.
That's so interesting to me and disgusting, but awesome that it's effective. And then my other question for you was, you were mentioning that there's fungi that is not present on the like plants, but that is present in the gut microbiome of the Drosophila. So yeah. I don't know if you touched on where that comes from or if I missed that or if we know that yet.
Yeah, that's a great question. So the first thing is that the fungi may be on the plants, but just in a really, really small quantity that we're not really picking up. Because we tend to, so when we collect these data, we do what's called a normalization process, which is microbial taxa, like fungal groups that have only a few DNA sequences that are spit out by the whole algorithm, that those tend to be not included in the final analyses. And there are a number of statistical reasons for that, which go above my head. But it could be that those fungi are present. They're just present in really low quantities, but that the flies are seeking them out differentially. Maybe they really love those fungi, and so they're eating them even though they're not common. Or it could be that those fungi are being picked up from something else other than the plants, or maybe other parts of the plants, because we were only sequencing the leaves of the plants, and it could be that the fruits of the plants or some other part of the plant has those fungi. But that's a great question. We're not totally sure.
That is so interesting. Thank you so much. And then I guess one more question for you. Um, when you mentioned that when the flies are not having enough microbes to be able to reproduce, I wonder if they were to get their microbes back up to a level, would they then be able to produce eggs, or have you not really seen that change? I guess like long term.
Yeah, no, that's actually an excellent question. Because we had some concern that maybe when we're knocking out the fungi of lab flies, we're using an antifungal chemical. It's the type of antifungal chemical that people spray on apples, common supermarket apples, to knock down fungi and keep them from rotting in the store. So we're giving that to the flies, and there was this question: is that antifungal actually toxic to the flies itself, and are the flies just not making eggs anymore because they're sick? So what we did is we did a whole nother set of experiments in the lab where we removed the fungi from fly guts using that antifungal, but then we took the flies off the antifungal. We started just feeding them normal food again that didn't have the antifungal in it, and we actually then inoculated them with microbes from flies that had never had the antifungal. And it turns out those flies, that we call that a rescue experiment, and the flies that we rescued in that way actually were able to then turn around and make eggs again. So we don't think the antifungal was acutely toxic to the fly. We do think that that's evidence that it's the fungi themselves that the flies do need to make the eggs. And so yes, you can recover that in a fly if it ends up getting the fungi back.
Oh, that's so awesome. Thank
Yeah,
you.
it was a very exciting experiment actually.
And I guess if no one else has any other questions, I am kind of curious, when the flies are eating in their natural environment, would they be eating more so like the live leaves, or do we know if they're more so eating the decomposing plant matter? Because it just makes me think of when they're attracted to like the rotting fruit and all of that, that maybe they would be attracted to like the decomposing plant material
Yeah,
too.
you're absolutely right. They are almost certainly mostly eating the decomposing plant material plus the microbes that are on that decomposing plant material. We also think that they're oftentimes just eating microbes right off of a healthy leaf without actually eating the leaf itself. But yes, largely they're getting most of their calories probably from yeasts and sugars that are in like rotting fruits of the different kind of plants. Yeah.
That is so cool. That just makes me think that they love like fermented foods like kimchi or something. It's like their equivalent.
Yep, I think you're absolutely right. Yeah. Yeah, and a lot of those things are probably healthy for us because they probably help foster a good set of microbes in our own guts. Absolutely.
My cutty, that's awesome. Thank you so much for sharing with us today. And if no one else has any questions, I think we're going to end soon. But I do want to say if anyone wants to follow up with Matthew, you can reach out to us and we can connect you. And then our next webinar is going to be in January, and we're going to be featuring Philip Meyer, and his talk will be on meteorological observations in a lava tube opening and the effects on vegetation. So mahalo, Matthew. Thank you so much for sharing with us today. And thank you everyone who was able to attend.
My pleasure. Thank you, everybody. Thank you, Tristan.